Lambda by 1 by 74 has 538 nucleotide bacteria. For lambda has 48502 base pairs. Extracellular quality is 4.6 into 2.6 base pair, and haploid content of the is 3.3 into 10 to the power 9 base pair. Let us recapitulate the chemical structure of polynucleotide change DNA or RNA. Nucleotide has a three components: a nitrogenous base, pentose sugar ribose in case of RNA, and deoxyribose over DNA, and a phosphate group. There are two types of nitrogenous bases: purines, adenine, and guanine, and pyrazine, cytosine, uracil, and cytosine is common for both DNA and RNA, and thymine is present in uracil. Uracil is present in RNA at the place of thymine, and nitrogenous base is linked to the OH of pentose sugar through an N-glycosidic linkage to form a nucleus such as adenosine, adenosine, or deoxyguanosine, cytidine, and deoxycytidine, uridine, or deoxythymidine. When a phosphate group is linked to OH of 5C of a nucleoside through phosphoester linkage, a corresponding nucleotide to a deoxynucleotide depending upon the types of sugar present response. Two nucleotides linked to through 385 the phosphodiester linkage to form a dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer thus formed has one end of free phosphate moiety at 5' end of the sugar, which is referred to as 5' end of the polynucleotide chain. Similarly, at the other end of the polymer, the sugar has free OH of 3 group, which is referred to as 3' end of the polynucleotide chain. The backbone of polynucleotide chain is formed due to sugar and phosphate. The nitrogenous bases link sugar moiety project from the backbone. In RNA, every nucleotide residue has an additional OH group present at 2' position in the ribosome. Also in the RNA, the uracil is found at the place of thymine 5-methyl uracil, another chemical name for thymine. DNA as an acidic substance present in the nucleus was first identified by Frederick Mitchell in 1869. He named it as a nuclein. However, due to technical limitation in isolating a long polymer intact, the elucidation of the DNA remained exclusive for a very long period of. It was only in 1953 that Thomson was an influential skill based on an X-ray diffraction data produced by Morris, Wilkes, and Rosen, and Franklin proposed a very simple but famous double helix model for the structure of DNA. One of the languages of the proposition based period between the two strands of the polynucleotide chains. However, this proposition was also based on the proposition of Erwin Chargaff that for a double-stranded DNA, the ratio between adenine and thymine and guanine cytosine are constant and equal one. The base pairing confer a very unique property to the polynucleotide chain. They are said to be complementary to each other, and therefore, if sequence of base in one strand is known, then the sequence on another strand can be predicted. Also, each strand of DNA, let us call it as parental DNA, act as a synthesis of a new strain. Two double-stranded RNA, let us call them as outer DNA thus produced would be identical to the parental DNA molecule. Because of this, the genetic implication of the structure of DNA become very clear. The salient feature of the double helix structure of DNA are as follows. It is one of two polynucleotide chains where the backbone is constituted by sugar, phosphate, and a base project inside. The two chains have anti-parallel polarity. It means if one chain has polarity 5-3 days, under has 3 days 5 days. The bases are two strands paired to the hydrogen bonds forming base pair. Adenine form two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded to cytosine with three hydrogen bonds. As a result, always a parent comes opposite to pyrimidine. This generates a approximately uniform distance between the two strands of the helix. The two strands chains are coiled in a right-handed fashion. The pitch of helix is 3.4 nanometer. Nanometer is 1 billionth of a meter. That is 10 to the power 9 meter. And there are roughly 10 base pair in each. Consequently, the distance between the base pair in a helix is approximately 0.34 nanometer. The plane of the one base pair stacks over the other in double helix. This, in addition to H1, confers stability of the helical structure. Proposition of double helix structure for DNA and simplification explaining the genetic become evolutionary very soon. Francis Crick proposed that the center of the molecular biology with this the genetic information flow from RNA, DNA, RNA, protein, in some case flow of information in reverse, that is from DNA, RNA to DNA. replication. While proposing that the double helical structure of DNA, Watson and Crick had immediately proposed a simple replication of DNA to require their original statement. This is as per it is another chapter noted that the specific pairing we have related immediately suggests a possible copying mechanism for the genetic material of Watson and Crick 1953. The seam suggests that the two strands would separate and act as template for the synthesis of new complementary strands after the completion of the replication. Each DNA molecule would have one parental and one newly synthesized strand. This seam was termed as a semi-conservative DNA replication. The experimental proof it is now proven that the DNA replication conservative was first shown in a colony higher organisms such as plants and human cells. Matthew Masselson and Franklin Stahl performed the following experiment in 1958. They grow E. coli in a medium containing NH4Cl and 15 is the heavy isotope of nitrogen as the only nitrogen source for many generations. The result was N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule would be distinguished from the normal DNA by centrifugation in a cesium CHCl density gradient. Please note that N15 is not a radioactive isotope and it cannot be separated from N40 only based on density. Then they transferred the cells into a medium with normal NH4Cl and took samples at various different time intervals as the cells multiplied and extracted their DNA remained as the double stranded helix. The various samples were separated independently on CHCl gradient to measure the density of DNA. The DNA that was extracted from the culture one generation after the transfer from N15 to N14 medium, that is after 20 minutes, E. coli device at 20 minutes had a hybrid or intermediate density DNA extracted from the culture after another generation, that is after 20 minutes, two generation was composed of equal amounts of this hybrid DNA and of light DNA. Various similar experiments involving use of radioactive remedy to detect the distribution of newly synthesized DNA in the chromosome was performed on Basia faba by Taylor and colleagues in 1958. The experiment proved that DNA in the chromosome also replicate. The living cells, such as the E. coli, the process of replication requires an onset of catalytic enzyme. The main enzyme is referred to as DNA-dependent DNA polymerase. It uses the DNA tablet to catalyze the polymerization of deoxynucleotides. These enzymes are highly efficient enzyme as they have to catalyze polymerization of a large number of nucleotides in a very short time. E. coli that has only 4.6 in 10^6 base pair. Compare it with human whose diploid contains 6.6 in 10^9 base pair. Completes the process of replication within 8 minutes. That means the average rate of polymerization has to be approximately 2000 base pair per second. Not only do these polymers have to be fast, but they have to be catalyzed the reaction with high degree of accuracy. Any mistake during replication will result into mutation. Furthermore, energetic replication is very expensive process. Deoxyribonucleic triphosphate several dual purposes. In addition to acting as substrate, they provide energy for polymerization and two terminal phosphate in the deoxynucleoside triphosphate, a high energy phosphate, same as in ATP. In addition to DNA-dependent DNA polymerase, many additional enzymes are required to complete the process of replication with high degree of accuracy. For long DNA molecules, the two strands of DNA cannot be separated in an entire length due to very high energy requirement. The replication occurs within a small opening of the DNA helix referred to as replication. For the DNA-dependent DNA polymerase, catalyzed polymerase only in one direction, that is 5 to 3 days. This creates some additional complication and replication. Focus directly on one strand, the template with polarity 3 days 5 dash application and continue while on others the template with polarity 5 dash this content. This continuous synthesized fragment are later joined by the enzyme DNA ligase. The DNA polymerase on their own cannot synthesize the process of replication. Also, the replication does not randomly at any place in DNA. There is a definite reason of E. coli DNA where the replication originates. Such regions are termed the origins of replication. It is because of the requirement of the origin of replication that a piece of DNA needed to be propagated during recombinant DNA procedure requires a vector. The vector provides the origin of replication, but not every detail of the replication understood. In equity, the replication of DNA takes place at S phase of the cell cycle. The replication of DNA and cell division should be highly coordinated. Failure in cell division after the DNA replication results in a polyploidy chromosomal anomaly. Transcription. The process of copying genetic information from one strand of the DNA RNA is termed as transcription. Here, also the principle of complementary governs the process of transcription, except the adherence of the complements new forms base pair with universal institute of Taiwan. However, unlike in the process of replication, which on onset in the total DNA of an organism get replicated in transcription, only a segment of DNA and only one of the strand is copied into RNA. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed first. If both strands act as stamps, they would code for RNA molecule with different sequences. Remain complementary does not mean identical. And if they code for protein, the sequence of amino acids of the protein would be different. Hence, one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Second, the two RNA molecules, if produced simultaneously, would be complementary to each other and would form a double-stranded RNA. This would prevent RNA from being translated into protein, and the exercise of the transcription would become a futile one. A transcription unit DNA is defined primarily by the three regions: the promoter, the secretogene, and the terminator. There is a convention in defining the strength of the DNA in the structure gene of the transcription unit, since the two strands have opposite polarity and DNA depends on the RNA polymerase. Also, catalyze the polymerase in only one direct, that is 5:3. The strand that has polarity 3 is as example and also referred to as template strand. The other strand in which polarity 5:3 and the sequence same as RNA except thymine and the place of uracil is displayed during transcription. Suddenly, this strand does not confer anything referred to the coding. So all the reference point while defining a transcription unit made with coding strand to explain the point a hypothetical sequence from a transcription will represent ATGTAC. The promoter and terminator flank the structure in a transcription unit. The promoter is said to be located towards the 5' and upstream of the structural gene. The reference is made with respect to the polarity of the coding state. It is a DNA sequence that provides binding site for RNA polymer and is the presence of promoter in a transcription unit. They also define the template and the coding strand by switching its position with terminator. The definition of coding and template strand could be reversed. The transcription is located toward 3' and downstream of the coding strand, and it is usually defined the end of the process of transcription. There are additional regulatory sequence that may be present further upstream or downstream to the promoter. Some of the properties will be discussed.